Everyone, welcome to the STORE lecture series. It's really my honor to introduce Jill Banfield as our STORE lecturer today. Jill received her PhD from Johns Hopkins University and is now a professor at Berkeley. She began her career in the geosciences, but went on to do some pioneering work using and developing metagenomic techniques to characterize microbial communities in acid mine drainage systems, and now works in many other environments. Her work has led to the characterization of many new lineages of microbial life and ultimately a revision of the tree of life itself. Jill has won numerous awards for this work, including being elected to the National Academy of Sciences and many other, a fellow of many other societies. And so please join me in welcoming Jill.
Hey, thank you for the invitation. Thank you, Rachel, for organizing the visit. It's a pleasure to be here today. I'm going to tell you about a part of our research which is really focused on what we call candidate phyla. So I want to take a moment to explain to you what I mean by candidate phyla, as written here. These are large chunks of the tree of life that are not represented by even a single isolate. And to this day, they've been known based on gene surveys, such as 16S RNA gene surveys, which basically tell you there's something there and something about where it places phylogenetically. But more importantly for me, methods that we broadly describe as metagenomics, but specifically the form of metagenomics that recovers genomes from microbial communities. So I'm going to start out with just a very brief description of the methods that we use, and then I'll really focus completely on the results that we get from applying these methods to a variety of different ecosystems. So basically we go into the environment and we sample an entire microbial community. We extract the DNA from the community, we fragment it, we sequence it, we then assemble the sequences to make larger fragments, and then we try and work out how to put those fragments back together in the right groups or draft genome bins, and ultimately in some cases we go in and we manually curate the bins, ultimately in some cases to a complete closed genome. The vast majority of cases, no, that's not the case. We're working with an approximation of the genome bin, and we basically focus our analyses on things that are about 90% complete, based on our best estimates. So this is a really important method, I think, because it allows us to get genomic sequence information and to predict the physiology of organisms that we otherwise know nothing about, other than perhaps their 16S RNA gene sequence. Because we sequence the entire community DNA, we also get genomes from phage, bacteriophage that are reproducing in bacteria, viruses of archaea, eukaryotic viruses, and even, in fact, most recently, the genomes of eukaryotes themselves. Mostly we work with microbial eukaryotes, but we can also get pretty decent genomes from a lot of other different kinds of eukaryotes as well. So I'll start off telling you about the voyage that I personally took, beginning with acid mine drainage, which you heard mentioned a moment ago. We worked, when my lab was in UW-Madison in Wisconsin, we used to come here to California and we would swing by Berkeley. Actually, we'd use Norm Pace's lab at Berkeley at the time as a stopping off point, and we would drive up to Redding in Northern California and we would go underground, deep into old mine tunnels and collect samples of biofilms. And the biofilms looked pretty much like what you could see there. They were floating on the surface of acid solution. pH 0.7, 0.8 typically, very, very acid. Green from all the metals, mostly iron, also a lot of copper and zinc. These were beautiful assembled microbial communities. And we began our work by doing 16S and then fluorescence in situ hybridization. And we did learn a little bit about how these organisms associated with each other in microbial communities. And we were pretty excited about this because we were able, for the first time, to get draft genomes from what we now call metagenomic data sets, and to predict something of the metabolism of bacteria, in this case shown in the top, and archaeon in the bottom. But that was only part of the story. We realized by examination of the fragments of genome sequences that were constructed, and at that time were quite low abundance, that there were other things in there that we were not detecting by 16S and thus by probes, by fish. These organisms were initially called something, then they were renamed to Arman, and we realized that they had very small genomes, very, very novel, couldn't work out really where they placed phylogenetically at the time, but ultimately came to understand that they were probably a pretty distant group out here on the tree of archaea that was known at the time. And based on their very small genomes, we deduced that they were probably pretty small cells, and in fact, we're able to establish that that is in fact the case. And so working with Luis Comolli, who was then basically a postdoc researcher at Lawrence Berkeley National Lab, we did a lot of microscopy, and most of it was cryo-electron tomography, and tried to characterize these communities in a cultivation-independent way. These were whole biofilm samples, but targeting specifically the identification of tiny cells. And these, then, are what we deduced to be the Arman archaea. You can see here a single cell with some ribosomes showing up in this slice of a tomographic image. So this is a three-dimensional data set that's been sliced. And here we see what was basically the dominant archaea in these biofilms. It's one of the thermoplasma talis, and it has no cell wall, so we know pretty much what it is based on a relatively restricted overall community composition. And we can see here these beautiful dividing cells attached to the surface of the Arman. So we deduced that these are episymbionts, organisms, archaea, living on the surfaces of other archaeal cells. So let me put this in context of what was known some time back now of the tree for archaea. Here we have the well-known and well-recognized archaea. Don't worry about the names. It's basically the major groups. And in the red boxes, we can have this group, which we are now calling the microarchaeota and parvarchaeota, basically the Armans from acid mine drainage. And next to them, we have the most famous of them all, Nanoarchaeum equitans, an amazing organism that Carl Stetter's lab was able to grow in mixed culture with its host Ichneococcus. And so this is cited here. So now we went from one peculiar episymbiont nanoarchaean to two. Here again, the Armans growing in association with the thermoplasma talis archaea, and here we have Nanoarchaeum growing with its host, again as an episymbiont. A little bit later on, Eric Allen went to Scripps in Southern California, and we teamed up with him to go out to Lake Tyrrell in Australia and study hypersaline microbial communities. We were interested to see if the methods that worked in acid mine drainage could be scaled up to more complex communities, because this is the time now Sanger sequencing became more available and then ultimately transitioned into Illumina. And lo and behold, after sequencing some of the samples, we start to realize that there's these really small genomes in there. And it turns out that they are in fact nanohaloarchaea, and they've now been recognized around the world. So this is a paper by one of Eric's postdocs in conjunction with ourselves, and then subsequently a cryo-TM study by Karen Andrade from our lab. So now we have three. Then Christian Rinke and others at the Joint Genome Institute, working with Tanya Wojcicki, began to sequence single cells from lots of different environments. And they realized that joining our Armans and the nanohaloarchaeota and the nanoarchaeota of Carl Stetter, we have two more groups. We have the I can't say this name, I'm not going to try, and then this one, archaeota. And they decided to take the first initial from each of the letters of the names and call them the DPAN. So the DPAN are archaea. They appear to potentially be a monophyletic group comprised of nanoarchaea. Around about the same time, we were doing some research at the Rifle site in Colorado. It's a DOE research site, looking at aquifers, groundwater and sediments. And we started to realize that coming out of the metagenomes were genome sequences for nanoarchaea, more nanoarchaea, but this time two major new groups. Actually, there's multiple major new groups, but two that were named, the Pace archaeota and the Woese archaeota. And so we can see we're now starting to expand this radiation within domain archaea with pretty divergent lineages. And so here, these are the DPAN, and this is then the acronym renamed with these additional groups. So now we say the DPAN are the original groups plus a bunch of others. And actually, Cindy Castell managed to sequence and assemble a lot of genomes and analyze them from the point of view of their physiology, and I'll talk about that in a moment. But she made this tree, which was published now some years ago, and just to get you a bit of a perspective what we're looking at here, this is a phylogenetic tree of archaea. There's the root to bacteria. From here, we go to basically all other archaea, and here we go from the root to the DPAN. So this is starting to look like a pretty substantial slice of the archaeal tree. More recently, Tom Williams, working with Martin Eberle, did a phylogenetic analysis with much superior skills than we could possibly have been able to bring to the task, and was able to make some conclusions regarding the monophyly of DPAN. I do think this is still an open question, but these authors concluded our analyses support the monophyly of the DPAN and place the tree root between DPAN and all other archaea. So this is making this group a pretty interesting group, I think. So some of the other work that we were doing at the time was at a site in Utah called Crystal Geyser. It's, oh no, because I can't project this, you don't get to see the geyser erupt. You'll have to imagine it's erupting up there in the corner. But this geyser draws water up from hundreds of meters below the surface in the Colorado Plateau, tapping into 3 different semi-discrete aquifers. So we basically collect samples night and day, night and day, night and day through what's called the eruption cycle, a period of time where the geyser erupts and then goes quiescent and erupts again. It's about 5 days. And based on the geochemistry, working with Catherine Ryan at the University of Calgary, we were able to link microbiology of these waters, these aquifers, to the depth where the aquifer derived. And so basically this is an overview of the community composition, the entire community. A group I'll talk about in a moment of bacteria, and I'm focusing now for the moment on the DPAN here. So you can see they change in abundance pretty substantially over the eruption cycle. And in particular, you can see this one very abundant archaean that based on its genome, we conclude, again, it's a nanoarchaean. Another one that's very prominent in the aquifer, just to give you an illustration of the metabolism side of things, is this one which has subsequently been named Forterra. I'll tell you why in a moment, but basically, I don't expect you to understand, because it's too small, the overall view of the DPAN, but we again infer that it is a symbiont on a host archaeal cell. It lacks many, many core biosynthetic capacities, and among other interesting things, it lacks basically all of upper glycolysis and basically the pentose phosphate pathway. What it does, it seems, is scavenge nucleotides and feeds them into a pathway called the AMP salvage pathway, the nucleotide salvage pathway, which is a CO2 incorporation step that uses rubisco. I'll come back to rubisco in a moment, because this is a bit of a theme. But basically, from the reconstruction, we get a sense of what this organism can and it can't do, and it can't do a lot. I did want to include this slide for another reason, and that is because it is possible to curate genomes to completion. And what's even nicer, maybe, is that particularly for bacteria, but in some cases archaea, you can actually confirm that the genome is probably pretty much correct. And that takes advantage of something that's been known from the various early days of genome sequencing, GC skew. So if you look at this, it has the typical form of GC skew starting at the origin to the terminus and coming back to the origin. This is actually not a very common form of replication in archaea to my knowledge, but it was described by Patrick Forterra. And Patrick has gone on record as being not a big fan of genomes from metagenomes, but I said to him, Patrick, I'll show you everything you want. I'll give you the genome, I'll give you the reads, I'll sit down with you and tell you about this, but I want to name this after you. And he said yes. So I hope he's changed his mind, at least in this case. So back to the eruption cycle. We can see here that there's a big spike in abundance of this particular named DPAN, right at one particular part of the eruption cycle, and if we compare its abundance to what we think might be, the host archaeon, we can infer but not prove that the host may be this Candidatus altarchaeum, SM1, which is another uncultivated candidate phyla, but known now by quite a few genomes. So it's a hypothesis generator at least. And so coming back to the question of where these organisms are originating, we find that during the eruption cycle, which is plotted on the timescale here, this is the same timescale, this period of high abundance of DPAN correlates with accessing water from the deepest aquifer. So these organisms are living in the deepest part of the subsurface that we've been able to sample, highly enriched in the fluid erupted at that time, which I want to go back here one second. So also coming up at the same time in the eruption cycle are these other group of organisms which I have not yet introduced you to, but I'm going to tell you now are bacteria, and we're going to call them the CPR. And I'll explain the acronym in a moment, but basically, well, it means candidate phyla radiation bacteria. So Those are very interesting to us and had been for some time, because working with Kelly Wright as a postdoc in our lab at the time in the Colorado Plateau rifle site, we sequenced acetate-emitted groundwater, and we found that the groundwater contained a huge number and variety of bacteria with very small genomes, probably very small cells, and minimal metabolic capacities. So the genomes are usually less than 1 megabase in length, and about up to half of the genomes have no discernible function based on anything you can do with bioinformatics. They do have core metabolism. They have a few interesting geochemical things. This one in particular has the ability for hydrogen and some sulfur metabolism, but they do not have an electron transport chain. They do not have more than a few or a single or maybe none components of the TCA cycle. And actually, they can't make their own lipids, to the best of our knowledge. They cannot make their cell envelope. So very, very minimal metabolism. One might think symbionts that depend on a host for provision of basic resources. Here's another example of one of these organisms that turned up in the group. This is another bacterium, also from a candidate phylum. This one wasn't even known from 16S surveys, and that's actually been pretty much a pattern that many of the genomes we're getting from phyla that actually weren't known even by 16S. But this is just a schematic of the cell, and if you look at the things, we've got these little red arrows through, you can see again they lack a lot of really basic core metabolism. They again cannot make their own lipids, and in this case, they have rubisco. Not all, but some, a few, have rubisco. And this is very interesting to us because this was now finding of a rubisco that previously was only known in archaea. So this is the AMP salvage pathway rubisco, or a variant of it that I mentioned previously. What they probably have is a fermentation-based metabolism. They're probably anaerobes, and they probably depend on other organisms for basic things, just like the DPAN archaea. Some have been found in protists, but the vast majority of the ones we're studying, and we're studying them at many different sites now, probably depend on bacteria, and I would guess, based on everything we know at this point, that they are also episymbionts, symbionts on the surface of other bacteria. Not a well-studied type of symbiosis at all in bacteria. And this is a pity because I can't use the animation, so I'm just going to step you through this. So we've hypothesized based on the fact that the genomes were consistently really tiny. There must be small cells. And so we conducted a repeat experiment of the experiment we published previously, but in this case, we collected cells onto the 1.2 micron filter. Then we let the water go through and we collected onto a 0.2 micron filter, and that's where most people stop. That's the sample they take into the lab. Big mistake. Because a lot of stuff goes through a 0.2 micron filter, and they are cells. And those of you who are microbiologists will probably recognize this filter as the filter used to filter sterilize. So what should come through? Definitely not cells. But cells came through, and we collected them. We cryoplunged them on site and brought the samples back to the lab and studied them with cryo-TM. This time it was work by Birgit Loof and others. She was a postdoc in our lab at the time, and was able to get some beautiful images of a variety of different cell shapes with cell features. What do we see? The cells are really tiny. They have, in this case, lots and lots of pili on the surface, and I'll come back to the topic of pili, but I think they're a critical feature of these organisms. So, because we have tomographic data, we can calculate exactly the volume of these cells, and it was determined that they were about 0.009 cubic microns. Now, that doesn't probably mean much to any of you, but actually, I want to tell you that that's the theoretical minimum size for life. It's not below, it's right at the theoretical size calculated by others. So, coming back to the cells here, the second pathway, we're now going to take those filtrates, post 0.2 micron and the 0.2 micron. We're going to shotgun sequence them and reconstruct genomes. And by doing this in a targeted way, we were able to recover a huge number of draft and near complete and a good handful of complete genomes. And this has been published in a variety of different places by different people. So Chris Brown was a PhD student in our lab at the time, worked on this collection of genomes, and he published a paper with almost 800 genomes from this radiation, of which you can see here actually 8 are complete, and the average genome complete was estimated at 91%, so pretty good. with this kind of sampling, you can be pretty sure what is and isn't present within the genomes, at least on average. Again, they have tiny genomes, and they fall together in what appeared to be basically a monophyletic radiation within domain bacteria, similar to what we saw with the archaea. You can see some names around here, but there's the CPR shown with a bunch of names and acronyms which reflect their original designations when they were detected in clone libraries, 16S. So because we saw this as a monophyletic radiation and all the radiation was basically candidate Phyla, we just described it as the candidate Phyla radiation. And I want to say this is not meant to be like a name, it's just a description of a physical feature of the tree. These are fascinating genomes, I would say transfixing. First of all, they lack what are normally considered to be central critical ribosomal proteins. Everything has to have this ribosomal protein or that one, and they would lack them, and consistently across entire huge groups of these organisms. For example, they lacked L1, some lack L9, whole big slices of the tree, and all of them lack, this is not so rare, but lack ribosomal protein L30. So this is very intriguing, considering basically what these organisms have is their information system, and that's what really distinguishes them from viruses, for example, or phage. We have begun to get TEM images again from these organisms. These are coming from a site called Genasi it's in the Central Valley. It's under a dairy farm, actually. And Chris is now a postdoc in the Kate Lab at Berkeley who studies ribosomes by TEM. And so they're going to basically collect tomographic data sets and resolve the structure of these ribosomes by most sophisticated methods. We're also really interested in areas like this. So just to give you a bit of context here, this is ice. And the scale bar here, this is a huge cell, would go right up way into the roof of the building. And there's one of these, what we believe to be CPR cells. And if you look, you can tell this cell knows there's something there because the wall is thickened and changed. This is a low resolution preliminary image, but we're very keen in the future to move this back to tomography and work with experts to really understand what's going on between these organisms. They have a lot of other interesting and intriguing features. One of them, very rare in bacteria, as far as we know to date, is that they have introns in their tRNAs. And very interesting introns too, but I'm not going to spend any time talking about them. But it is a feature that was not commonly found in bacteria, but reasonably common in various CPR. This is just showing you pictures of the tRNAs, and this is showing the excision sites, and then you can see two different cases here where the intron is excised from the tRNA. They have lots of other cool things. Some people might think this is the most cool. I'm not sure I agree, but everybody really likes CRISPR-Cas these days, yes? Yes? Okay. They have really interesting CRISPR-Cas systems, and much to my embarrassment, actually, we first reported the sequences from CPF1, but didn't really say anything about them. We just said that the CPR in general have very few CRISPR systems, and when they do, they have kind of weird ones. And certain MIT Broad people who will go unnamed. But we don't have anything going on with patenting in Berkeley and those guys. I'm not going to say anything more about that. But anyway, they're enriched in the CPR, and then more recently we reported with Jennifer Doudna's lab Cas-Y, which is a new type 2 system which has some significant potential for genome editing coming from the CPR. So small genomes, compact systems may be important if you're interested in genome editing and genome editing technologies. But they have a lot of other cool things. I didn't mention the introns in the rRNAs, but that was one of the big findings. Initially, and then working with David Valentine's group, we've reported these things called diversity-generating retroelements, which are really rare, but they're just really prominent in certain regions. And some groups, Phyla I should say, have repurposed the stop codon, one of their stop codons, to code for an amino acid, so alternatively coded. And they have heaps of hypothetical proteins. I'll talk more about the proteins of CPR in a minute, but I wanted to come back to the DPAN and the CPR. So I don't know if you've heard what I'm saying and you get my gist, but basically these are two huge groups within bacterial and archaeal domains, and they have a lot of features that seem pretty similar. So Cindy Castell put together this beautiful diagram. It's way too complicated. This is actually a lot of information that shows you what capacities are present in the DPAN and CPR. And all I want you to take away from this is it's sort of like a mirror image. But beyond this, beyond the very similar capacities and deficits in the metabolism, they have a lot of features, and some features, at least in CPR, that aren't meant to be in CPR. They're not meant to be in bacteria at all. They're normally archaeal things. And likewise, there are things in the deep pan that normally are only found in bacteria. We're just starting to really get a sense of what those are. But overall, we have these two similar stories of putative symbionts, small genomes, probably mostly episymbionts, from major groups that have been almost completely unstudied. Why are they unstudied? Can't grow them in pure culture. I mean, it's, I think, a reflection of the fact that these are obligately dependent on other organisms for basic resources. And actually, that's what really interests me about microbial communities, interactions. So this is a really obvious case of an interaction. It could really be quite important in terms of impacting the function of microbial communities in nature. What do we think they're actually doing? Well, because they can't make so much stuff, they're getting it from other sources. They may be scavenging material. And we hypothesize that the CPR and DPAN existing in the deep subsurface, for example, this is just sort of quartz grains, hang out down there and basically capture rare resources, this is a very nutrient-poor environment, and repurpose them back for use by other organisms. The scavengers, in a sense, in some part. But let me just put that in a slightly different way. This is a cartoon that Kelly Wright and others put together for another purpose, but it serves to illustrate some of what we're trying to do with this research. We aren't just describing individual organisms, we're trying to put the information together to understand a community. How do organisms work together to turn Earth's biogeochemical cycles? So here on the left we have resources. It's a very simple diagram. Refractory carbon, more easily accessed carbon going down. And we have organisms represented by these circles. We have products of their metabolism, for example, through fermentation, these compounds, acetate, lactate, hydrogen, ethanol, and so forth, that then feed other organisms in the community, such as the famous, from Rifleside especially, geobacter organisms, which carry out geochemically important transformations, such as iron reduction, as shown here, and uranium reduction. So there's a network with resources and organisms and products. But what's important here is we can now incorporate into these conceptual models and actual models information about organisms that have no cultivated representatives. So when I say that they're in the subsurface, may be important in recycling, I mean they're basically turning compounds, detritus from other organisms back into compounds that feed other community members. So, okay, we have the DPAN in the archaea. Deep divide may be monophyletic. We have the CPR in the bacteria. Maybe monophyletic. In our trees, kind of looked like they may be the deepest branch. We thought, well, nobody's made a tree of life before. Let's kind of put it all together. And what we really wanted to do is not worry about the eukaryotes at all. We just wanted to see, are they the deepest branches in both domains? This is a calculation that requires fairly intensive computation, and I want to give a special thanks to the team at the Cyprus Science Gateway because we made extensive use of their computational capacities to make this tree. Now, in this case, we concatenated the ribosomal proteins. These are genomes, but they're draft genomes, and we don't want to make chimeras, hybrids, due to binning errors. So we picked the ribosomal proteins because they're co-encoded on a single small piece of DNA. They're all together, consistently, bacteria and archaea. Okay, so basically this gives you a little bit of details, but there's a tree to come, and I'll share what we found. So this is the tree as it emerged from the analysis with some pretty colors decorating the various components. Here is the bacteria. Here are the archaea, and let's not talk about the eukaryotes. Actually, I got a lot of grief over this tree, believe me, because we supported the view, according to the people who didn't read the paper, that the eukaryotes branched within the archaea, which was a claim made by Thijs Edema, and I have been connected with him in publication a little, but that's his case. We actually say you can't support the deep branch order, so we're not making any claim here. So let's forget about the eukaryotes, focus on bacteria and archaea. What I want you to see is, first of all, the huge diversity of bacteria. Is this a sampling error? No, I don't think so. I think this is a true reflection because we're sampling whole communities and everybody pretty much finds bacteria are more abundant and diverse than archaea in their environments, with some exceptions maybe. Here are the CPR. The red dots indicate lineages that have no isolated representative, candidate Phyla. In this tree, again, the DPAN are not monophyletic, but actually if you take the bacteria out, they do become monophyletic. So it's still, as I said, an open question. So the big point we were trying to make with this paper is that huge amount of bacteria, a huge diversity within the CPR, the candidate phyla radiation, all the red dots, and the prevalence overall, not just here, but overall, of lineages that lack isolated representatives. And I wanted to just make a shout, because this is always referred to as the hug et al. Tree, but I just want to say this tree came from a lot of investments of a lot of people who I've listed here, including collaborators, David Relman, Ron Amundsen, Brett Baker, and so forth. So we pulled together a lot of sequences that were coming from genomes from metagenomes at the time. It has changed a little bit, but not very much. We recalculated it a few months ago. Just to give you a bit of perspective on these two branches again, here we have the CPR with another image from Birgit Löf's work showing a tiny little cell, and then the deep anarchia down here. So, I personally think that figure 1 of this paper is the least interesting figure, and I really like this one better. And I'll tell you why. This is the same tree. It's exactly the same data. It's just rendered in a different way. What we said is, let's forget about whether the actinobacteria phylum, whether the chloroflexia phylum. Let's just let the tree tell us where the diversity is to be found across the domains. Let's just collapse the branch lengths to an equal value across the entire tree. And you'll notice we've lost the eukaryotes, just chopped them out, threw them away. In black, we have branches with one isolated representative, and in colors, red for archaea, blue for bacteria, we have the candidate phyla. Here is the candidate phyla radiation. And if you look at that, the way I look at that, that's around about half of bacterial diversity. Well, needless to say, perhaps this was not a happy thought to a lot of people. And they've now come up with a number of statements that I think are very intriguing. This is all because the ribosomes of the CPR are fast evolving. I don't know. Maybe it's an artifact. Maybe they're symbionts, so they're fast evolving. There's nothing actually we can find in the genomes that would say that they're actively, currently undergoing rapid genome reduction. Honestly, I don't care too much about why, but that is a huge amount of diversity wherever it came from. But we were concerned about this question, and I'm going to show you next is unpublished data. We were concerned because, well, let's look at this from another perspective. So Raphael Mehust and David Burstein working together clustered all the proteins from a huge number of novel genomes from as balanced a view as they could get from, first of all, the new data, the tree basically. And they made this plot, and I'm going to explain it to you. It takes a little explanation, but basically we're going to ask the protein universe to vote. Are the CPR really that different? Do they hang together? Do they group with other symbionts in insects, for example, from Nancy Moran's work with reduced genomes? Or are they really distinct? So, every line here is a genome, and every column here is a protein family. In the entire analysis, there are 22,000 protein families. But here, we're just focusing on about 800 protein families that were determined to occur in modules or groups of proteins that are widespread. So we're not really thinking about the proteins that are found in one phylum, one genus, rare things. We're just focusing on the common protein families. Black is present, white is absent, and this is 800 things, this is 2,300 things. So we're really, really zoomed out here. But you can learn what we need to learn by just a glance. First of all, the genomes have been hierarchically clustered based on the pattern of presence or absence of these common proteins. Here, because our data set did include a lot of CPR, in this first analysis, we have the CPR clustering all together. We didn't make them cluster. They clustered themselves through this hierarchical clustering analysis. Here are other bacteria. These are proteins that are depleted in CPR, things they lack. Here's the information machinery, which, as I mentioned to you, is well preserved, pretty much, in the CPR. And here are other proteins. The first thing I want to do is just look at the hierarchical clustering. I want to say, what does the clustering tell us about relatedness of these organisms without reverting to a phylogenetic tree at all? There is no protein sequence alignment coming up. This is just looking at this hierarchical clustering and rendering it in a different way. So let's look at that. It looks like this. The insert for your reference in the top left-hand side is basically a calculation, a recalculation of the tree I showed you previously, and it's just to give you some bearings. Here are archaea. There were a few archaea in our data set. It's not our intention to focus on archaea here. Here are the non-CPR bacteria, and as I told you, our data set is enriched in CPR. and here are the CPR. Well, the first thing that we're really interested to find is that basically within the CPR we get a pretty strong phylogenetic signal, but a huge diversity in range of metabolic capacities. We'll come back to this view in a moment. Now I want to go back to this slide, and this is exactly the slide I showed you before, and I want to draw your attention to the proteins or the protein families on the right-hand side. What we learned from this is not only did the CPR lack stuff that could be a reflection of genome reduction, they also have a bunch of proteins that distinguish them from other bacteria that are found very, very rarely in what I'll call normal or non-CPR bacteria. So we're pretty interested in those. They're widespread, they're common proteins in the CPR, but they're not present in other bacteria to speak of. Just looking at this, of course, many of them are hypothetical proteins, but the take-home view to our best ability to determine at this present time is these are proteins involved in cell-cell interaction, including some really novel pili subunits and things to maintain the structure of pili. So this is, again, a zoom in of this. What you see here now is, again, this is hierarchically clustered. Things are grouping together pretty much, for the most part, based on their name, which is based on their taxonomy from phylogenetic trees. Interestingly, the TM6, which are not part of the CPR but look a lot like CPR, a good example of maybe parallel evolution, yeah, they look like it, but they do not have these CPR-enriched families, supporting the fact that they are in fact not part of the CPR. Well, we thought, let's have a look within the CPR in more detail, and I think this is a small but important point. The CPR are not monolithic. They're not all the same. We can see here just a quick snapshot of the center part with our 800 protein families, presence black, absence white. We can see very clearly here there are certain groups that have a lot of metabolic capacities, two classes probably for the Peregrine bacteria, and there are some that have very minimal capabilities, and this seems to be at the level of the major groups, maybe you would say Phyla. So that's intriguing. The next thing we wanted to do is just determine whether or not what we were seeing was an artifact of studying genomes from the aquifers. Does it make any difference? So Raf downloaded one genome per genus approximately from NCBI and made a phylogenetic tree, this is a phylogenetic tree, to show the organisms he chose to put into the analysis. Again, a few archaea, including some DPAN. A small number of CPR, relatively speaking, only 10% of the data set. And then a bunch of other bacterial phyla, and then these stars are the known, well-studied symbionts with reduced genomes. So the question is, do the CPR group with these symbionts? And the answer is pretty much no. So this is, again, the same kind of picture I showed you before, very zoomed out. Protein families, black present, white absent. Completely obvious what the CPR is in this diagram. These are the CPR. So one type, two types, and the rest of bacteria. So we would say this looks like a metabolic divide within domain bacteria, CPR and non-CPR. Nothing to do with phylogenetic trees. What's also interesting, but I don't think I have time to talk about it at all, is that if you look at the hierarchical clustering, again, just taking the clustering off this diagram and wrapping it around, we can see here archaea and DPAN separating nicely. CPR, the colors in the CPR just reflect some specific groups of interest, but green and red, and then all other bacteria. And what you'll notice is that in some cases, the correspondence between the presence-absence patterns of these common protein families is pretty strong in terms of their phylogeny. For example, there's the cyanobacteria. They all group very tightly. And it's actually this group here. So a strong signal that would seem to indicate that once these lineages arose, I'm speculating, that they kind of determined the metabolic platform or the backbone for whatever new evolutionary opportunity was available to them, and they persisted more or less roughly to this day. Not that they didn't get other things, but that core has been pretty persistent. And so one might think the same were true of the CPR, that they may have had this long symbiotic history associated perhaps with the co-evolution with the non-CPR on which they're dependent. But this is, of course, speculation. It remains to be seen. So you might be thinking, okay, who cares about the CPR and DPAN, right? They exist in the Colorado Plateau. I don't really care about them. They are actually pretty much found everywhere in the world. They're definitely less common in aerobic environments. The open ocean seems to be absent or deficit of them. But this is just a pretty picture that shows all the different environments and even apparently the space station. So this is an important take-home point. They are everywhere. But I also want to say that they are in the human microbiome, and I hate to admit it, but people really care about the human microbiome because they like humans, and that is a hot topic. So I want to tell you, yes, you cannot forget about them in the context of the human microbiome, though it's very much an open question, because I think I forgot to tell you that many, many phyla of CPR cannot be detected, and phyla of the DPAN cannot be detected with standard 16S primers. So first of all, we lose them with the primers, maybe 20% of them. And second of all, we lose them if they go through the filter we collect our sample onto. But they are in a lot of places, and they are now, and have been for a while, recognized in the mouth. And in a work with David Relman's lab, we found quite an interesting diversity in the dolphin mouth, which is this beautiful cover. They've been reported not through our work in the lung. And in fact, there's an intriguing paper from Stephen Quake's group that says they're quite abundant in a certain cohort of blood samples as cell-free DNA. We do not know where they're coming from, but I will tell you the pretty well-held guess is that it's possibly drinking water, because that's where they're very, very commonly found, water. And some have even reported that they're enriched in sterilized water. Okay, one very brief comment about soil. We're working with Mary Firestone, who's a professor at UC Berkeley. She's a soil scientist. She's trying to track carbon from plants into the soil. I'm not going to talk about the majority of that work, of course. I'm just going to talk about a work with a co-supervised student in which we tracked the 13 carbon that was fed to the plant into the rhizosphere through SIP, stable isotope probing, and then we did genome-resolved analysis of that data. So we separate the DNA and sequence it separately based on its weight. It's separated. So if it takes up the label, it should be heavier in the heavy fraction. We can work out which organism's taken up the DNA. Does that make sense? Okay. So basically tracking that. Anyway, so intriguingly, one CPR bacterium was in the heavily labeled category, implying that its DNA contained a lot of 13C. Well, that seemed very fun for about 5 minutes until we realized, well, wait a second. Like most CPR, this particular group cannot make their own DNA. So there's something wrong here, but actually it's something right, and I think very interesting. They cannot have taken the 13C and made nucleotides, but they sure could take nucleotides from bacteria, potentially, or fungi, or potentially even the plant itself, and repurpose that DNA for their own nucleotide. They can make polymers out of nucleic acids, they just can't make them. So we basically have a little story where the CPR is actually receiving the 13C, but more nuanced, given the fact that they're dependent on other organisms. So it's kind of a fun story to conclude on. So this is my last slide. I want to say that subsurface is a pretty cool place to work. It has been the environment where many, many of the new genomes and groups and lineages have been found. I did not talk about the Loki Archaeota, for example, but this work I mentioned by Thijs Eddemer and others, Brett Baker, looking at very, very interesting organisms that are coming from there that are also changing our view of the tree of life. I personally am absolutely transfixed by the CPR and DPAN, particularly as they may represent half of all bacterial and archaeal diversity. Don't know exactly yet, but that would be something worth serious consideration. They have uniformly pretty much small genomes, novel proteome compositions, unexpected metabolic features. I just scratched the surface, but they're really fascinating in terms of what they have and what they're not supposed to have, but they appear to have. They are likely episymbionts of other microorganisms, which, as I said, is a pretty much unknown lifestyle. And I think many of the proteins in their novel proteomes probably are involved in interaction with the host, because many of them are membrane-associated surface proteins, lots of investment in pili. But many other pathways as well, and I think it will be a rich data set for future exploration. And I'll just put this unpublished data on the table for you to think about. It does appear when we ask these organisms from the perspective of their proteomes, what are you, that the CPR at least, and I think the signal is there for the DPAN to represent a kind of metabolic divide in both domains. So I'm going to finish up with some acknowledgements. I've touched on work that's been published over many years. Most of it, though, I've really focused on has been recent, and so I acknowledge mostly the people who've been working on these projects most recently. Some of them no longer with our research group, now employed elsewhere. And I want, of course, to thank our funders, US Department of Energy, the IGI, Chan Zuckerberg Institute very recently, that's called BioHub, and then also the Sloan Foundation, which is supporting the Crystal Geyser Deep Subsurface Life work. And thank you for your attention. Jonathan.
Many symbionts co-evolve with their hosts in a variety of ways, either co-phylogenetics or they exchange genes with their hosts. Do you have any information that suggests that these have exchanged any DNA with other organisms that might be their hosts or that have sort of co-phylogenetics with them?
You know, we have looked, and there is not a very strong signal. I mentioned a lot of other people's work. I forgot to mention the work specifically by he et al. I did have it on the slide with the human. They showed by enrichment recently that they are symbiotic on actinobacteria, and I think that there may be others from that group who are also symbiotic on actinobacteria, but there isn't a really obvious signal in the genomes. Basically, the genomes, if you look at them, look really, really novel. And at first, they just look like nothing we'd ever seen. And as we have had more genomes, we find they look like each other, but not so much like a host. So it's definitely a good thought, and it's something we would continue to look for. Yeah. I think they attach to, and you're asking me a good way, envision, I'm just imagining. I believe that they are cellular entities. They have all the cell division stuff that they need pretty much. I believe that they either, and it's probably many different answers, either scavenge lipids, that's the big problem, the lipids, from the environment, or they scavenge them from the host. And given that we see now, in a number of occasions, the cell division happening when they're attached to the surface of another cell, some data I showed, some I didn't, I just think they must somehow pinch out a bit of the membrane of the host. But I would love to know. Yeah, we're really, really trying to get them into enrichments so that we can start to probe those questions. Yeah. Yeah.
Given the position of these little things, they must go back almost to the origin of life, or what we have of it. What do you think that means?
I don't know, obviously. I mean, we really hoped that we could find deep support, strong support for the deep position in that tree. But notice I didn't point that out because I can't. Because unfortunately that tree is not good enough to say that it's the deepest branch. But I do suspect it might be. I do suspect that they're pretty ancient, but they are symbionts. So I imagine this is how it happened, that once upon a time there was some primordial existence, and that over time the Earth changed and new lineages appeared with new capabilities, and that spurred the evolution in parallel of the symbiont. So I think that they are probably ancient. The counter to that would be, no, they're just all rapidly evolving. I honestly don't think that's the case, but we will see. It's hard to believe it's genome reduction over that huge a slab of the tree consistently. So I think they've been that way for a long time, and I think that's the story the rest of domain bacteria is telling us, that once established, these platforms persist mostly. Do you agree? So do you agree? Do you agree?
Well, it sounds like they must be around forever.
Around forever. Yeah. Is that another question back here? Yeah.
Do you think the metabolites are coming out into solution, or are they being transferred by some mechanism, cell to cell?
Okay, so one of the things that's really been hard to find are transporters. I'm sure they have them, very few transporters. They have pili. They have a huge investment in pili. We see the pili in the EM. I think they're using pili for a lot of stuff. That would be my guess. They also have a lot of surface attachment proteins, and they have huge proteins, thousands and thousands of amino acids, and sometimes in clumps in the genome. So really big investment in excreted proteins that are probably on the surface. So I don't know exactly how those are working. They have basically type 2 secretion systems. Yeah, so I don't know what they're taking up from solution. Yeah. Joe? Yes.
Does it have the signatures of the rubiscos we're most familiar with, or did it evolve to this one?
Okay, so I will tell you that Bob Tabita's group at Ohio State University complemented a strain deficient in rubisco from the CBB pathway, and it does function as ribisco. They are form 3 and form 2,3 ribiscos, otherwise known from archaea. We haven't actually gone and published that we think that some might have phosphoribullar kinase, but there's an indication that in some cases they may have something that looks like the CBB pathway, but mostly it looks like the nucleotide salvage pathway. But it is the same basic chemistry. Yeah. Yeah. Sorry, yes, you go ahead.
From their genomes, do you think they have anything that they're offering the cell that they're in symbiosis with, or do you think it's more of a sort of parasitic type lifestyle?
I think that radiation is so huge, everything goes. If you think of all the divergent things that bacteria that are not CPR do, I imagine there's equal diversity. In fact, I don't think I managed to remember to tell you that the branch lengths in the CPR from that rendition of the tree that's based on the protein presence and absence, where they're 10% of the data set, they're half of the diversity in metabolic platform. So they're really diverse in what they do. I think, basically, yeah, probably anything goes, but my speculation, and we did publish this somehow, is that you asked about the membrane. I think they steal the membrane from the host, and they basically sit on the surface and act as phage decoys. And that they use then, if they can survive, they have a lot of restriction systems. If they can survive, they basically repurpose the nucleotides from phage into the DNA. It's a fairy story, but it seems like a good one. Yeah. There was another question back there, yeah.
Yeah, I'm intrigued by the idea that they pinch off a little bit of material. Have you ever seen the genome of a nanobacterium inside of its host? In other words, is there a model there that maybe the genome is an entity that gets inside the host and then gets pinched off as a blab to move on?
I don't have any data to support that. They do exist as cellular entities within protists in a few cases, but not as naked DNA. And they have a lot of investment in cell stuff. They make peptidoglycan. They actually just can't make lipids. So they've got a lot of stuff that tell me that cellular at some part, and clearly they're cellular in some part of their life, but I really don't know. I would love to know, but I don't know.
And even though they're quite reduced in genome size, is it possible they also have some other non-coding DNA that would be atypical for these small genomes?
I think the coding density is pretty common, pretty similar to the normal coding density, also pretty much the same size. So nothing that stands out. They have, as I said, they have introns in their rRNAs and tRNAs, but those are pretty tiny. So it's not a big part of the genome.
Phage and Phyla make great attachment points for phages. Are you finding phages specifically associated with the CPR or more, or?
DPAN I know more about because we just did more microscopy, but yes, we find phage on the surfaces of the CPR and DPAN cells all the time. And they do sometimes stick to pili, as you know, so yeah. There's no question. It's been hard to find the phage of the CPR, but we do have some because first of all, they look like CPR from the point of view of their novel genomes, but also because we don't have the CRISPR systems to link them. So they do have phage, definitely. They do have prophage, but mostly we see evidence, as you suggested, from the imaging that they're predated by phage. Yeah. I don't think we've ever found like a secondary metabolite cluster, for example. So, I don't think we have any evidence that they make antibiotics. I'll tell you what they do make, and maybe you guys can help me on this. They make a lot of the CPR, a good number of the CPR have the mevalonate pathway down to the precursors, all the way through to the precursors for the lipids. I wouldn't be surprised if they're not using them to make some kind of compounds that would be involved in interaction. Or proteins? Sorry.
Non-ribosomal type of proteins.
Yeah. So that's why I said, they don't have the secondary metabolite biosynthetic gene clusters, no. I don't think we've ever found them. I don't think so, no. And we're looking for them, but just haven't found them. So, yeah, those are pretty big, those gene clusters, so it would be surprising maybe to find them.
So how far along the mevalonate pathway do
So they go to isopentyl PP, pretty far. They can make the tRNA that's dimethyl whatever it's called, tRNA, yeah. They can make that, which is the precursor for a plant, and a plant hormone, I think, yeah. No, nothing beyond that. We've looked at every single genome, some of these genomes, with every single thing we can think to throw at it, and that's basically all we can find. I think there's a lady back there, yeah.
Can you explain a little more how you can use GC skew to estimate genome completeness?
Oh, great question. It's not it's not genome completeness, it's genome accuracy. So basically, bacteria replicate, their origin determines replication, and they have a systematic use of G and C, so G plus C, and G minus C over G plus C is the GC skew. And usually the two replication paths are about the same length, not always the same, so we basically get this pattern of cumulative GC skew that looks like this. And actually, I'll tell you, I guess I can tell you, that we actually did an audit of NCBI's complete genomes, and just to show that GC skew is robust. And anybody who did genomics in the early days knows about this method. It's been around forever. We didn't invent it, don't get that idea. We just remembered it and use it. But actually, I'll tell you what it looks like when a genome is wrong. When it's right, it goes like this. When it's wrong, it goes like that. And actually, there's a good number of genomes in the public databases that do not have the correct GC skew despite being closed bacterial genomes. And actually, we can see where they went wrong, because there's repeats that you just have to flip. So it's actually really good for detecting assembly errors. First time we found this was in, I think, one of the well-known, I think it was Citrobacter coceri. It had 12 errors in the genome, because basically we realized when we looked at it that they just stuck the genome together randomly at all the rRNA operons. They just went, well, I don't know how this genome goes, and nobody will ever check, so they just stuck it together. And then when you look at the genome, GC skew, it's just like out the window. So we take it, when we get a genome closed and we've gone and checked everything looks really good, we run the GC skew and it looks like this, we go, one last stamp of approval. But it just does come from that basic replication process. Yeah. Yep, some of them have very low skew. Actually, the skew ranges enormously from almost nothing to in units 20,000, 30,000. Yeah, but the cyanobacteria are known not to have much skew. So when we don't see any skew, we don't get too worried. It's where we do see the skew and it looks rapidly wrong that we would want to look further. But it's a good point. They don't all show strong. It's still most do, though, overall, when you look at like 7,000, 8,000 genomes. Yeah? Yeah?
So, just out of curiosity, how long were the 16 proteins that were used in those maximum likelihood trees?
Oh, I think it's, the entire alignment is, I'm going to say 2,200 amino acids, 2,300 amino acids, something like that.
So the pathways for nucleotide salvage in both DPAM and CPR, are those in essence identical between the two, or are they more analogous processes?
Okay, so the pathway is not that well known even by people who study it, but there are a couple of enzymes in the AMP salvage pathway that are known, and they actually have well-known misannotations, and they're the same. Yeah, and they tend to be together in the genome, but the Rubisco is solid. I mean, you can really recognize the Rubisco. So it's the lack of the PRK and the other genes of the CBP pathway that basically first convinced us it had to be nucleotide salvage. And it makes sense for these genomes.
Clear to what degree they're similar to each other, just that they're both serving a purpose?
I think just generally similar pathways. I mean, I can't even start to tell you what a diversity of Rubiscos there are out there that you don't know about yet, but there's a lot. And within the groups are new forms coming up. But basically what we can say is they look like Rubisco, they're making good branches in the Rubisco tree, and they have these other enzymes that fit with that pathway. Question? Yeah.
So I was struck by the TCA side that you don't see. So I saw in your slides that you have the pentosphosphate side, right? The pentosphosphate.
Yeah.
Do you see both oxidative and the reductive portions of the pathway?
It varies a lot. But we just see the part that goes, we actually have to use the pentose phosphate pathway to get around one enzyme in the glycolysis pathway, just is routinely absent, so it's just got that bit.
Phase of the pathway would be for could be for nucleotides.
Yeah. Sometimes, and some of them can make their own nucleotides. It's rare. Some can. When I showed you a misleading example, I'm afraid, because I showed you the one case where they don't have the PPP pathway, but mostly they just have that one loop. And then sometimes they'll feed into nucleotide biosynthesis. Some basically have nothing, which is amazing to me. Yeah. That I know of.